Start with the seminar today. Thank you very much for coming another day. Today we have Aina Pons, who is a PhD student. Well, I guess almost everybody knows her. She's a PhD student at the Maris Group, and she's going to talk about monkeys. Actually, right, it's about fish and microbiome. So thank you very much, Aina. The first.
Thank you for coming today to the seminar. I'm on my last year of my PhD, so today I'm going to talk about a chapter of it. It's all about investigating the correlation between the social network and gut microbiome in a wild marine fish population. First of all, just to introduce, as you know, animals and humans have individual behavioral differences when compared with members of the same species and the same sex and age. And when these behavioral differences are consistent over time and across ecological contexts, we can categorize individuals as behavioral types. These behavioral types are widespread in nature across all the different taxa. And in fishes, we can summarize these interspecific behavioral differences through 5 major axes, which are exploration, aggressiveness, activity, boldness, and sociability. Sociability is, today I'm going to focus on this one, and sociability is defined as the reaction to the presence or the absence to conspecifics. Many fish are social, and these social interactions play an important role on the ecological and evolutionary processes on animal populations and at understanding the functioning of these ecosystems and their resilience to environmental perturbations. So how do we study these social interactions? Well, because animal observations can sometimes be very difficult to study in some species or even nearly impossible in many species, and traditional data collection techniques have limitations. We have animal tracking systems that allows us to monitor the movements and to obtain precise information from a large number of individuals at the same time. And with a high resolution telemetry system, acoustic telemetry systems, we can locate and obtain the trajectories of animals tagged with acoustic transmitters, and therefore we are able to reconstruct social networks of wild populations. So the microbiota gut-brain axis. In the animal gut holds diverse bacterial communities that can affect the behavior, physiology, and the metabolism of their host. In the gastrointestinal tract, or the gut, we have the gut microbiome, which is all of the collection of bacteria and their related genes that live in the gut, and they can produce some neuroactive chemicals that then can travel to the brain. So the gut is connected to the brain, specifically to the hypothalamus, which is a brain region that controls hunger and emotions. And this connection is made through the vagus nerve. So this is a bidirectional connection, and it's called the gut-brain axis. So the microbiota gut-brain axis links the endocrine system with the nervous system and the immune system. So you may wonder what is the connection between social behavior and animal gut microbiome. So this line of research is very recent. There are some studies. One example is with wild chimpanzee, where they found important connections between the animal microbiome and social behavior. The microbiome can affect social interactions by affecting the whole central nervous system and the peripheral chemical communication. And conversely, social interactions can affect the composition and function of this microbiome. But this connection in the wild is poorly understood, and there are a very few number of studies studying this connection yet. So our model species is the pearly razorfish, Xyrichtys novacula. It's we reconstructed the social network of this species, and it's a small coastal wrasse that inhabits sandy bottoms of Mediterranean Sea and Atlantic Ocean. It's a diurnal species and has a typical behavior. It buries itself in the sand during the night or to avoid predators. It's a protogenous hermaphrodite, has a carnivorous diet based on small benthic invertebrates. And here you can see the typical behavior when it buries itself in the sand. Wait. So this study was done between April 2019 and October 2019, encompassing the spawning period of this species. The study site was composed by sandy patch of 12.5 hectares with depths between 12 and 17 meters. The area was surrounded by Posidonia oceanica seagrass meadows, and we installed a total of 70 receivers in the area with a distance in between of 50 meters. So we used a high-resolution acoustic telemetry system based on the juvenile sun moon acoustic telemetry system manufactured by Biotech. These are the receivers and the transmitters that we used. And we tagged a total of 320 individuals with these sizes. So associations were quantified with a 5-minute interval, and we defined an association when two individuals were less than 1 meter away from each other. And for the gut microbiome, we took a subsample of the social network with a total of 26 individuals. We then extracted the DNA with a kit. And then we did the analysis of the 16S rRNA gene amplicons with a standard protocol for sequencing and using in the Illumina platform. So in overall, what we did was to analyze the social network. We created an association matrix. Then for the microbiome, we identified and investigated the overall microbiome diversity, the core microbiome diversity, and we created a species abundance dissimilarity matrix for investigating the relationship between sex and microbial composition. We did a principal component analysis and ANOVA, and finally to explore the possible correlation between the social network and the gut microbiome, we did a mantle test. So the results. First of all, for the social network, we obtained a high-quality positioning data set of 7,000 one-day-long trajectories. And this is very interesting, we found a harem-like social structure. Here you can see like every patch is a single territory of one individual, and in males you can see in green you can see the males, and in pink the females. And territories are formed by one male and several females, and males display agonistic behavior towards their neighbors. Here you can see the reconstruction of the social network of the 200 individuals. And here you have the trajectories in a specific day. And there are other species tagged in this area too. Sorry. So for the overall microbiome diversity, we obtained a total of 547,000 high-quality gene sequences with a total of 1,019 OPUs. We found only 2 species that were present in all of the individuals. One was Ralstonia, with an average relative abundance of 16%. This was the most abundant species. And the other species was Ruegeria, with an average relative abundance of 1.5%. And the second most abundant OPU was Clostridium, with a 4% average abundance relative. Then we looked for the core microbiome. The core microbiome can be defined as a set of the bacteria and their related genes that are characteristic of a specific host or environment and could have an important function. And here you can see the relative abundances of the core microbiome that we found. You can see that the most abundant were Ralstonia and Subdoligranulum. About the correlation between sex and gut microbial composition, here this is orange, but here it's not very well seen. But you can see that the males have more variability in terms of the gut microbial composition compared to females. But when we did the ANOVA, we didn't find significant differences between sex and gut microbiome. For the correlation between the social network and gut microbiome, we performed the mantle test. We looked at the Breit-Curtis distances, and then we looked at the social network associations at 60% of occurrence. And we found significance. And what is the interpretation? Well, gut microbiome diversity is correlated with the association of the social network. This means that individuals that have a shorter distance, that are from the same harem, present a more similar gut microbiome diversity. So high throughput tracking allows us to reconstruct wild living marine fish populations. The taxa identified resemble those found in other fish species, as Proteobacteria being the dominant phylum. All the fish in this only 2 species were present in all of the individuals and could have some important functions. And individual microbiome samples of fish from the same harem were more similar to each other, while differences to other harems were strong. One possible explanation for this is that there could be some transmission pathways between the individuals that are at a shorter distance or are in the same harem. We see a correlation between the social network and the gut microbiome, but the mechanisms that play a part in these relationships remain unknown and may involve social transmission and shared environmental factors like diet. For further studies, we propose increasing the number of individuals, control for different environmental factors such as diet, and trying with different time series, for example. And to conclude, this study is among the first in wild marine fish and highlights the importance of the potential transmission pathways that can play a part in this social gut microbiome. And this highlights too the potential implication this may have for wild animal populations and the evolutionary and ecological dynamics of social behavior and ecosystem functioning. Thank you.
So, thanks for the talk, by the way. The diet bit, I think, is I find it quite interesting because if I remember well, no quote on that. Humans living in the same house, they tend to have similar microbiome because they share essentially food and other things. Okay,
they share the same environment.
Much bigger, but in your case, if I understand well, your fish, it's like small crustaceans, right? Or
yes, they eat mainly mollusks and crustaceans, like spring.
So, would you expect that the bacteria associated with these would be significantly different across the different parts of your sampling site, so that they could justify the differences in what's the name of that? Inter-harem, thank you. Gut microbiome.
So, if I understood you well, you mean that in a small area of our area, we could have a different diet, and this can
right. So if you
can make the difference,
that
or
has different bacteria associated to it, which has probably has a spatial structure similar to well, at least the one in the harems, then you could get fish that have the same kind of gut microbiome because they feed on the same
yes, but we assume that the area is small, so they feed on the same prey. So there is no quite a difference in the diet.
Have you checked that? Meaning, if I get a mollusk from here and from there, is the difference in microbial microbes associated with the prey, let's say, lower than the difference between the gut microbiomes?
We didn't have control factors like diet, so this is maybe a good idea for the future to see if there are some different preys and maybe compare different areas, not just only one, because maybe if I put you an example. Maybe there is a population in, I don't know, in the south of Mallorca, and maybe if we do a study between this same population of this study and the population in the south of Mallorca, we find some differences, and maybe it's because they have a different diet. Because but in this case, we can't say that is caused because of the diet.
Just a related question. Because they bury themselves in the sand, right? So
yes.
Do you know how uniform is the set of microbes living in different patches across? You will not know, but maybe something is known about how diverse, how patchy is the distribution of the profile of species across the different sediments.
Yeah, we haven't checked for the bacterial species that live in the sediment. Maybe it can play a part, but we don't know. There are a lot of things that we don't know. We are just trying to understand the baseline, and then you.
I didn't understand how do you define the core microbiome from the total microbiome that you can find in the gut of an individual? How, what are the criteria you use to select?
Yes. There is a lot of discrepancy with the core microbiome and how it's defined. So there are studies that this core microbiome, maybe they put a threshold in a minimum the 30% of occurrence, and there are other studies that use the abundance. We used the occurrence, and we set our threshold in 60%. So there is a lot of controversy, and it's not very well defined between. Sorry?
Possibility that these microorganisms perform a specific function for the animal. It's only based on the abundance of the occurrence.
Yes.
Okay.
The occurrence, not the abundance. If it's present or not in an individual. Sorry.
Yeah, you were talking at the beginning about sociability.
Yes.
Yeah. Maybe I missed it later on, but is it possible to quantify sociability in individuals or differences?
This is well studied in controlled conditions, like laboratory conditions, where you have a set of aquariums and you do different tests to test the behavioral types, like sociability, for example. But in the wild is more difficult. So what we did was to test this sociability by looking at the interaction between individuals and put a minimum distance to see if there is an interaction or not. And this minimum distance was 1 meter.
So was there any correlation between the sociability and the gut composition or biome?
If
you found any, yeah, I mean, because at the beginning you were saying that there is this kind of feedback, no, between gut composition and sociability. So maybe I missed it, but if you have some kind of way to quantify sociability, was then related to the composition in the gut or not?
You can have a lot of ways to look at it, but we tried to see if in an harem the gut microbiome was more similar than other harems. I don't know if you understand it, but
I was just wondering if there was some kind of species that would increase sociability or not.
Yeah, there are studies that that found that some specific bacteria are present in more social individuals, but we didn't look at it, so I can't say which bacteria play a role in these interactions.
Thank you.
I was going to ask a very similar question. You said that the microbiota affected or could affect several things like the behavior or the health. So that was my question, whether you checked whether it any impact the diversity or the specific composition of the microbiota on something like behavior that I understood not, but what about the fish health, the size or I don't know what is important for the fish.
Yeah, I understand. We have, well, I have another chapter of my PhD with Sphaerosa aurata, Xyrichtys brim, and yeah, we looked at the composition and diversity and the structure of the microbiome, and for the 5 axes that I presented, and we found some differences in the structure of the microbiome in terms of co-occurrence of a species. They co-occur different depending, for example, if they are more active animals or not. Yeah.
That's very interesting.
Yes, I have another comment because I understand where you are saying that the particular composition of the microbiota can affect the behavior of the fish, apparently, or is it a mutual relationship? Because I was wondering, if I assume also that the diet is important, no? Maybe
Yes, it's very important.
maybe fishes that have a particular behavior that they make them be more aggressive or more efficient in predation or capturing specific organisms that might control the microbiome they have. So it would be not a consequence, so the behavior won't be a consequence of the microbiota composition, but the behavior determines what would be the microbiome through the specific particular mixture of prey they can able to catch.
Yes, interesting. Yes, the mechanisms are not well studied yet, but yeah, there is the question if it's the microbiome that controls some part of the behavior, or if it's the behavior that controls the microbiome and changes the composition. So this is a very good question and very
But I
good.
wonder if in any other species, this question has been addressed or not.
They try to address, but it's very difficult because you have a lot of factors that interact in this interaction. So it's very hard to understand.
More questions? Okay, so we can leave it here. Thank you very much, Ina.